with the recording. I guess you will have to accept, but this seems to be running. Okay, so it's 303. Sorry for the hiccup with the login.
That's
I
okay.
hope people
we understand it's your first time.
But this seems to be fine. So I think, Debbie, you could start with your introduction and sharing the session. And so welcome, Chloe, and good luck and enjoy.
Okay, awesome. So it's my pleasure to introduce Chloe Schmidt. So Chloe is currently a postdoctoral fellow at the University of Manitoba, working with Colin Garraway. She just finished up a postdoc at Yale with Walter Yetz, and she did her PhD with Colin in Manitoba and her master's at the University of Lausanne. So she's in a little bit homegrown. So Chloe is going to talk about macrogenetics and her work with kind of urban macrogenetics. And if any of you have any questions, she'll stay on afterwards for the question session, but you can also email her or me for that. Okay, Chloe, please take it away.
Thank you, Debbie. And thanks, Debbie and Valentina, for organizing everything and inviting me to come give this seminar today. This is really exciting. So I'm going to be talking about environmental and also human social drivers of urban biodiversity at both genetic and species levels. So, as I'm sure we all know, cities are the world's newest habitat type, and they are growing really quickly. So over half the world's population currently lives in cities, and for the 30 years between 1985 and 2015, urban land area expanded by over 80%. And this rate of urban land expansion has actually now cities are becoming, one of the major causes of habitat loss and fragmentation for wildlife. And this is clearly something that's of conservation concern. And so cities, a city is a city kind of wherever you go across the globe, right? So there's a few key processes that are kind of involved when we're talking about urbanization in general. So some of these we'll tend to see, of course, in addition to very high human population density in cities. In terms of environments, we'll see increases in impervious surface cover, so paved roads and buildings. Also increased temperature, so the urban heat island effect, where the temperatures in urban cores tend to be higher than in surrounding environments. There's also an increase in invasive species in cities, and these tend to be kind of pest species that are associated with humans and human movement, as well as a lot of habitat fragmentation that occurs. And so this has resulted in an ecological homogenization in cities across the globe, to the extent that actually cities tend to be more similar to each other in terms of environment than they are to their nearby natural surroundings. And this ecological homogenization is also accompanied by biological homogenization. So at this stage, we're pretty well aware that cities tend to harbor lower species diversity, as well as decreased functional diversity and phylogenetic diversity. But kind of at these broad spatial scales, there's a lot of data available to work with species distributions as well as their traits. And so we're able to test the effects of ecological homogenization on these kind of higher levels of biodiversity. But we really didn't know much about the distribution of genetic diversity in cities and whether there are these same kind of broad scale trends. So can we say that genetic diversity tends to be lower in cities, kind of, wherever you are on the globe? And so we have expectations about how urbanization should affect genetic diversity. So as we move across an urbanization gradient, what we expect to see is kind of increasing habitat fragmentation. And this is hypothesized to be a main driver of consistent effects of urbanization on genetic diversity, and specifically neutral genetic diversity. So the idea is that as we move from these kind of large, well-connected populations that live in natural habitats, we start to see kind of populations that are becoming smaller and more fragmented. And so to the point where you are in cities, wildlife populations here are going to have a more difficult time kind of moving around the remaining habitat patches available in cities. And so we're expecting to see decreases in effective population size, which means stronger effects of genetic drift. So we expect to see decreases in genetic diversity and also increases in genetic differentiation, because as I said, gene flow is more difficult within cities. And so this is kind of the working hypothesis of what we think is going on in cities. Obviously, this is closely tied to habitat fragmentation and also movement ability of organisms. So while we generally expect to see decreases in genetic diversity, the extent to which this occurs could also be related to different species traits that determine their ability to move around cities. And so the first general question that I tackled here and that I'll be speaking about here today was just a kind of simple test of this hypothesis to see whether urban wildlife populations actually tend to have lower genetic diversity. And I was able to do this taking an approach that's kind of now being called macrogenetics. And macrogenetics really is, taking advantage of the huge amounts of genetic data that are available in public digital repositories. So macrogenetics is essentially kind of, big population or landscape genetics. So it's tackling population genetics questions at very broad spatial, temporal, or taxonomic scales. And macrogenetics is now possible because, evolutionary biology was one of the first disciplines to really embrace this culture of open data and data sharing. And so there's been genetic data accumulating online for a very long time now. So there's a lot of it just freely available. And now that we're improving our access to metadata associated with these genetic data, we're able to explore patterns of genetic diversity in a spatial context at scales that weren't previously possible. And so with macrogenetics, there's a lot of different ways to get the data. So they could be harvested from the literature using summaries of genetic data. But my approach was to bring together raw genetic data. So I'm working directly with, genotype data, specifically from microsatellites. So during my PhD, I started to assemble a database of microsatellite data sets with which to address these types of macrogenetics questions. And so the data set is still growing, but what I'm going to be talking about today was work that was mostly accomplished during my PhD, which focused on genetic data sets that were sampled in the US and Canada. And so to amass these data, I did a systematic search through Datum, which is a platform that allows access to multiple digital repositories. Most of this genetic data was stored on the Dryad repository, if you're familiar. And at the moment for Canada and the US, we've got data from over 100 species of terrestrial vertebrates that were sampled at around 1,700 sites across the continent. This in total is sampled from over 60,000 individual animals. And so here I focused only on native terrestrial vertebrate species. Most of the data comes from mammals, but we've also got birds, amphibians, and reptiles in here. And so each of these points on the map represents a single species that was sampled at a single site. And now, because I was working with the raw genetic data, that enabled me to be able to estimate genetic metrics that I was interested in, regardless of whether or not they were presented in their original publications. And so that's really one of the strengths of using raw data for macrogenetics purposes. And so for each site, I estimated the contemporary effective population size, which is an indicator of the strength of genetic drift acting on a population, 2 different measures of genetic diversity, so gene diversity and allelic richness, and finally, population-specific FST, which is a measure of population differentiation. And so for my first question here, to really understand whether there are general trends in genetic diversity patterns across urbanization gradients, I also quantified the extent of urbanization at each of these genetic sample sites in 3 different ways. So the first one was just a binary categorization. So I classified each site as being either urban or rural based on census data. The second, I measured the human population density at each of the sites. And finally, the third metric of urbanization was the human footprint index. And this is a continuous measure of just kind of human habitat disturbance in general. So it ranges from 0 to 100, where 0 is the most natural and 100 is the most transformed or most disturbed environments. And human footprint index was especially nice for these purposes because it's kind of a more comprehensive measure of urbanization. In addition to human population density, it also takes into account other factors like roads, railways, nighttime lights, access to navigable rivers, and a lot of different aspects. So yeah, this was the most comprehensive measure of kind of habitat disturbance caused by humans. And so to actually test for the effects of these 3 measures of urbanization on our metrics of genetic composition, we used Bayesian hierarchical models, and kind of the defining feature of these models was to include both random slopes and random intercepts. And this allowed us to account for different average levels of genetic diversity across species, as well as species' different responses to urbanization. So, you know, if a mouse was more extremely affected by urbanization than a bear or something, for example. And so this allowed us to estimate species-specific effects of urbanization on genetic diversity, as well as an overall general effect from where we'd be able to say that, oh yes, you know, wildlife tend to be negatively affected by urbanization. Okay, so moving into the results now. First, I'll go through the results for mammals. And here we're looking at a coefficient plot for the overall model coefficients. That's on the x-axis, and then the y-axis, we have our 3 different urbanization metrics. The genetic metrics are here in different colors. The open circle is the parameter estimate for the effect of the urbanization metric on the genetic metric, and the bold and narrow lines are 90 and 95% credible intervals. So when those intervals overlap 0, that kind of means the effect is ambiguous or there is no effect. So looking here for mammals, it was super interesting to see. Our predictions were actually quite consistent with the results that we saw. So we can see that for all different metrics of urbanization, we're tending to see smaller effective population sizes and lower genetic diversity with increasing urbanization, as well as greater population differentiation in more urban areas. But then we look at birds and it's kind of a mess. So here we can see that our credible intervals are all pretty wide and overlapping 0, and there's nothing we can really say about how urbanization is affecting genetic diversity in birds in general across all the species in our sample. Looking at amphibians, we're seeing basically the same thing. Again, these very wide, credible intervals that are mostly overlapping 0. But funnily enough, for our two continuous measures of urbanization, so human population density and human footprint, we're starting to see a trend kind of in the opposite direction of what we expected. So higher genetic diversity in cities and lower genetic differentiation. But looking under the hood at the species-specific effects here, it really seems that these kinds of positive trends are due to very strong positive effects in only 1 or 2 species. So this definitely isn't something that's generally applicable to amphibians. And so kind of taking all of these results together, we can see that there's quite a bit we can see that there's quite a bit of nuance in the ways that wildlife genetic diversity are affected by urbanization. And kind of, just brainstorming about what could be causing this variation across these different taxa, we're thinking it could be related to movement ability, kind of like I explained earlier. So for birds, we didn't see strong responses in general across species, and it could be that their ability to fly actually allows them to maintain quite large, well-connected populations in cities. And this is in contrast to mammals, where we saw consistent negative effects, but of course, kind of land-bound mammals are going to be more greatly affected by barriers to movement and fragmentation in cities. On the very other end of the spectrum, we have amphibians, who are probably the worst at moving out of all of these taxa, and they're also quite sensitive to their environmental conditions. So we think that the reasons we were unable to detect genetic effects for amphibians could be that local populations are actually just being completely extirpated before we're able to detect any effects on genetic diversity. And so I just want to point these out quickly. The last word on this study was to looking at the actual species-specific effects that are underlying the models I just showed. And mammals and birds in particular were interesting because we can see for the mammal species, we've got the 0 line highlighted in orange here. Every single species had a negative response to urbanization. And so this is why we were seeing those very, kind of distinct negative responses to urbanization for mammals. On the other hand, if we look at birds, we can see quite a few of them do have negative responses to urbanization, but there's also a couple that tend to have higher genetic diversity in cities. And so it's this kind of spread of some species having negative results and other species having positive results that kind of averages out to no overall effect across these species. And so in the future, I think it'd be really interesting if we can build up our sample size for birds and kind of really explore traits or something that could be causing this variation in their responses to urbanization. So everything I've talked about up until now, we're looking at variation across urban to rural gradients. And this is kind of treating cities as if they, could be in some kind of environmental replicate, in a natural experimental setup. But that fails to take into account the fact that, cities aren't all exactly alike. Cities are different sizes, they're different ages, and there's a lot of potential variation that can come from just, specific histories of cities or the ways that humans are choosing to structure and develop their cities in different places. And so just as an example, there was a recent study that just came out looking at adaptive declines in clover to urbanization gradients. And so here they were looking for gradients in the expression of cyanide in clover, which is an anti-predator defense, with the idea being that in more rural areas, you have more predators, and so you're going to see higher expression of cyanide, and then that's going to grade out as we move closer to cities. And so they did this really cool global experiment, you know, sampling clover across the globe. And they actually found that decline did trend in the direction they expected for, like, I think about half of the cities in their sample, which I think is really remarkable to be able to even pick that up. But on the other hand, there's still half of the cities that either didn't exhibit this decline or actually exhibited a negative decline. So that's suggesting that there's still some, you know, differences between these cities. You know, maybe some cities do have higher herbivores, you know, maybe it's more rabbits or something like this. And so I think this is a good example of how cities are very alike, but also there's still environments and environmental differences that are causing variation in these species trends that we're expecting to see. And so I collaborated on a study that was led by my colleague Rika Kinnunen that was specifically exploring these differences between cities and how they support different, in this case, bird communities. And so here we focused on looking at how the traits of cities can filter for bird species with different life history traits. And so we looked at three different aspects of cities. So first was a city's compactness, and this was, you know, simply a measure of population density. So the city's population size divided by area, as well as median income of an entire city, and just the straight up population size of the city. And the bird traits that we looked at were kind of just like key life history traits. So body size, clutch size, and longevity, as well as migratory status. And here, again, we're looking at kind of the same coefficient plots I showed before. But we can see that the compactness of the city, and to some extent, some of the other aspects of cities, but they weren't that strong. Compactness had pretty notable effects on filtering for life history traits in birds. So cities that were more compact tended to have smaller species that had shorter lives and had larger clutch sizes. More compact cities also tended to, species were more likely to be migratory in more compact cities. And these results were particularly interesting because, you know, when we think about gradients in life history traits, gradients and declines in life history traits in natural environments, one idea is that these are related to resource availability. So in areas where you have few resources, we're tending to see larger species that prioritize offspring quality over quantity. And so potentially something about the compactness of a city could be modifying resource availability in a way that is selecting for, or filtering rather, for species that have kind of fast life history traits. And so this was, you know, an exploratory analysis looking at this trait variation, but it's something definitely to dig into further. And it again, you know, proves the point that there are variation in cities can have important effects on biodiversity. And so taking this a step further, I then wanted to look at this from a genetic perspective, of course, but here focusing on not only variation across different cities, but also looking at differences within cities. So how neighborhood differences can also be affecting genetic diversity patterns. And this study was inspired by this really nice review paper that came out a couple years ago that was addressing the effects of systemic racism on ecology and evolution in cities. And a very interesting idea that this paper brought up was the fact that, at least in the US, the racial composition of a neighborhood could be a quite suitable proxy for habitat quality of a neighborhood. And this is primarily due to a policy that was enacted in the 1930s in the US, which we now kind of colloquially call redlining. And this is when different neighborhoods of cities were ranked according to their desirability. So, you know, is it a nice area to live? And then racial and ethnic minorities were then systematically excluded from purchasing homes in these areas. And redlining basically created a socially structured urban environment in the US where these higher graded areas tended to have, more tree cover, older canopy cover, more green space, more parks, more yards and things compared to these less desirable areas. And even though redlining was eventually outlawed in 1968 with the passing of the Fair Housing Act, we do still see these trends in urban environments in the US today, which is really quite sad. And today, so we still see in areas that are predominantly minority communities, they tend to live in places that have very high population density. They have more urban infrastructure, so they're more intensely developed with buildings and roads. And we also I lost my train of thought. But yes, so essentially redlining was kind of pushing minority communities to live towards urban core areas that are more polluted and more densely urbanized. And so we thought that these kind of environmental relationships and the ways that they're associated with race in the US could be producing environments that have differential support of wildlife populations. So we would expect that in these areas that were highly rated during redlining would be able to support larger wildlife populations because there are more natural resources available. So they would be able to support these kind of larger populations that are more well connected. And so to test this, I used the genetic data from all 4 terrestrial vertebrate classes. And I took only sites in the United States that were located within cities. So we're no longer looking across urban to rural gradients here. We're only considering sites that are urban. And I filtered out non-urban sites, again, using this layer of census designated urban areas. And then for each of these sites, I quantified the proportion of residents that identified as white. And I used this metric because the goal of racial segregation in the US was to kind of maintain homogeneous populations of people who identified as white. But there's, of course, a lot of racial variation in different regions in the US. And so were we to kind of focus on these different races, we might see a lot of mixed results. And then to examine the relationships between racial composition at our sites and to see whether our expectations about environmental differences were consistent with what we know about the effects of redlining. I just made a scatter plot here, and we can see that there is a quite strong negative correlation between the proportion of white residents in a neighborhood and the human footprint index, which, as I mentioned before, is kind of a comprehensive measure of just general environmental disturbance caused by humans. And so here we can see still that communities that are primarily composed of minority residents tend to have higher human footprint index, so they're more environmentally disturbed. And so going from this relationships that we could find amongst our urban sites, we then expected we would be able to detect differences in genetic diversity and differentiation. And so again, we're looking at these very familiar coefficient plots. The only difference here is that now behind the overall coefficient estimates for the effects of proportion of white residents on genetic composition, all these little bubbles are the underlying species-specific effects, and the diameter of the circles are our sample size, or the number of sites we had for that species. And we can see here that our hunch was indeed correct. So in areas that have higher proportions of white residents, we're tending to see larger effective population sizes, higher genetic diversity, as well as lower genetic differentiation. And something that's notable to point out here was that in different taxa, when we were comparing genetic diversity patterns across urbanization gradients, as I mentioned, there are these very variable species-specific effects for birds and amphibians, where we weren't able to detect effects, and the consistent effects were only seen for mammals. But it actually appears that when we're only considering urban populations and looking at a very specific urban variable, and here a measure of segregation, we're tending to see generally consistent effects across all of the vertebrate classes that we included in this analysis. So give or take a few outliers, it really seems that kind of once we're in the city, all wildlife classes are affected in similar ways by these kinds of broad indexes of habitat quality that we see in cities. Also, I'll note that racial segregation explained a very small amount of the variation in genetic diversity. And this is kind of to be expected because our random effect here was doing a lot of work. So we're accounting for variation in genetic diversity across species, but then also across different taxonomic classes. So the fact that we see small effect sizes was to an extent expected, but then there's also this kind of temporal aspect to consider because cities are such new environments. It's very unlikely that populations there are going to be in mutation drift equilibrium. And so it could be the case that we're currently underestimating these effect sizes because we really have yet to see what the full consequences of urbanization and segregation and other factors that are affecting the distribution of habitats in cities. We could also maybe look at this from the bright side and say that these effect sizes are quite small, so maybe there's still time to turn this around, or at least kind of slow down decreases in genetic diversity by more equitably distributing habitat across neighborhoods in cities, and again, particular to the US. But I think this is a case study to say that the availability and connectivity of green spaces and other natural resources in cities is really important for maintaining high levels of genetic diversity. And so these results here, unfortunately, just kind of add to existing evidence that minority communities in the US are really bearing this burden of kind of living in more degraded habitats, but now also having kind of less access to biodiversity, which, bad for biodiversity, but that's also very bad for human mental health. And so knowing this and kind of then moving forward, building our cities and developing them in different ways will have benefits for both wildlife and people. So just to wrap up and bring all of this together, the initial question was to look at whether, for genetic diversity, we could see these general trends in response to urbanization like we do for other aspects of biodiversity at kind of higher levels of biological urbanization. And to an extent, yes, we can make some generalizations. So, we can say at least for mammals, genetic diversity tends to be lower in cities, but we can't extend these generalizations very easily across other taxa. And so that's something to consider when we're talking about, potential management implications or using evidence for kinds of, conservation actions, that not all taxa are responding in the same ways. We also saw differences when we're comparing populations across urban to rural gradients from only looking at urban populations, which is another very important consideration because, even if, for example, we can't detect general effects in birds. That doesn't mean that urbanization doesn't have an effect on birds, and it doesn't mean that by improving the habitat quality in cities, it's not going to have an effect, because as we can see from only looking at urban populations, that doesn't seem to be true. So in general, the takeaway from all this, in my opinion, is that really anything we can do to improve habitat quality and connectivity in cities, so both within neighborhoods, in cities, and also with surrounding natural populations, will be for the benefit of maintaining genetic diversity in wildlife, and hopefully maintaining adaptive potential, so that wildlife are more resilient and better able to cope with rapidly changing conditions in cities. So that's it. I want to say thank you to my supervisor and lab mates at the University of Manitoba, as well as to all of my wonderful collaborators who helped with this work. And finally, of course, to all the authors out there who post their data in public repositories, because without them, I wouldn't have been able to do any of this work. And thank you for your attention, and I'm happy to take questions.
Great talk, Chloe. Thank you so much. That was so cool. So yeah, I'd love to open up the floor to anyone's questions for Chloe. You can either raise your hand or post them in the chat. Oh, I see Alex has raised his hand. So why don't you unmute yourself? And I can't see everyone. So otherwise, if you can raise your hand virtually, but Alex can definitely go first.
Actually, I'm being sneaky. That's Alex there.
I know, but I was.
Yeah, cool, great talk. I love this massive data aggregation that you've done. It's awesome. You see all these massive data sets kicking around, and then actually pulling them all together is pretty awesome. I like how you've done it from scratch as well.
Thank you.
Cool. Yeah. So the question was, so you kind of aggregated, or kind of looked, I think, yeah, especially the first paper you're talking about, the 2021, and you're kind of looking at the effects on the different classes of species that you had. Have you dug into looking at like the effect of kind of life history traits or like dispersal ability and stuff, instead of looking at like just class? Could do like, yeah, dispersal traits and how that affects these different genetic metrics you've calculated?
Yeah, so nothing concrete to say right now, but I'm working with a student now to kind of dig into the effects on birds specifically, because just eyeballing those species specific plots quickly, it kind of looks like it was birds that are somehow associated with water tended to have decreased genetic diversity in cities. So it was waterfowl, but also, shorebirds and things like this. And so that seemed like a weird and interesting trend. And I half wonder if it's just because there's not a lot of habitat for these birds in cities. And so maybe that's why we're seeing what we're seeing. The student also suggested that maybe the water birds don't like fly as well in cities, I guess, because they tend to be larger. We've got a lot of geese and ducks in there. But yeah, like we're obviously going to include traits there that would be related to dispersal distance. Something else interesting was, of course, we had some bats in this data set, but they also responded negatively to urbanization. So I think it's not kind of a simple relationship with only dispersal distance once we kind of start digging into the differences within classes, but yeah, so we're working on it, but
yeah, no, sorry, it's just an interesting how you see so much variation within classes, and then there must be so many other different variables involved. Yeah, just wondering how far you got into pulling that apart. But cool. All right, thanks.
Felix?
Hi, Chloe. Thanks for this cool insight into this urban pattern. I wonder if using such metadata, or no, not the metadata, but doing a meta-analysis type of study on published data could also compromise your findings, because different studies will have applied a completely different sampling scheme. And I don't know how you did it, whether you really classified into rural, urban, or gradient also. So take a decision which is urban, which is rural, might also add to the uncertainty of the data. So what is your feeling about that?
Yeah, I think there are still sources of uncertainty, depending on where people might have chosen to sample the data and kind of the structure they use to sample them. But I think kind of compared to meta-analysis or a strict meta-analysis where you're just taking effect sizes from other studies, macrogenetics using kind of raw data synthesis really has the upper hand, because we're already eliminating all the variability that's associated with kind of pulling together different metrics of genetic diversity that might have been calculated in different ways, but also we're able to kind of quantify our environments in a systematic way across all of these sites. And so to classify sites as urban or rural, I really just use the census has shape files that designate city boundaries kind of according to their own internal designations. And so there wasn't really anything on my part kind of deciding what should be urban, what should be rural. It just did a merge a shapefile with the data and kind of put that together. And so I do think that that's a real strength of macrogenetics. And also because we're working with these data sets that weren't specifically sampled to address questions related to urbanization, that also means we're not getting any biases in the types of species that people would choose to look at with regards to urbanization and things like that. So yes, there's definitely noise, but I think taking the synthesis approach rather than a very kind of strict meta-analysis, that was able to overcome a lot of that. So not to babble on too far, but for example, there's another genetic database out there that compiled microsatellite genetic diversity, but used summaries from the literature. And so there, for metrics like allelic richness or observed heterozygosity, there is a kind of, there's more input on the data collector's part for how to quantify those. So observed heterozygosity is going to be really strongly affected by the different sample sizes across sites. And so that's why we chose to use expected heterozygosity instead. And also our allelic richness is kind of standardized to the same minimum number of individuals across the entire data set. So that's a comparable metric. Whereas if you're using allelic richness, that was, each standardized to a specific data set's minimum number of sampled individuals, then that's kind of less comparable and adds noise to the data.
Okay, thanks.
Christian?
Yeah, probably I continue on that quickly. What do you mean with raw data in microsites? So it's genotype tables, what you actually collect?
Yes. So it's not the rawest form of the data, but it's the genotypes. It's usually, data you would put into like structure or something. So formatted to that extent.
But that means if you have like two studies of species A, you don't combine them, because then you have again the problem that they are binning the alleles in different ways and so on. So actually your sampling unit is not the species, but the study still.
Yes.
Yeah.
So yeah, in that case, I would say all the species, we obviously can't combine data sets from different studies because most often they chose different loci to sample. But we only had multiple data sets from very few species in here, which was kind of surprising. But some of the more popular species like deer and coyote, we'd have 2 or 3 data sets maybe, but by and large, it's a single species and a single study.
And one more question, is the data set publicly available, or did you have to publish it with the publication, or is it?
Yeah.
Everybody can exploit it now, or?
Yes, you can. It's at least for the published studies that I presented here, the data is on Dryad. It's not, you know, the exact data, because Dryad doesn't let you publish coordinate locations for endangered species, but those were few in the data set. So yes, still currently expanding it, so the entire data isn't available yet, but the North American data are.
Thank you. It's very impressive, I have to say.
Thank you.
So Chloe, I had a quick question going to amphibians. So during here in Europe, I've heard from amphibian researchers that human created ponds are actually becoming the most frequent type of habitat for European amphibians. I don't know if that's the case in North America, and I was wondering if it is, and if that might explain why you see this positive effect to some extent with urbanization.
Yeah, funny you mentioned that, because that's kind of exactly what we were suspecting for those few species that had strong positive effects. I don't know if human-made ponds are kind of where you see amphibians the most in cities in the US, but I know that it is very common. And also, you know, roadside ditches and things like that, a lot of pond breeding amphibians really love. Honestly, amphibians are kind of tricky in a lot of ways. So, you know, we've done a lot of other work with these data, looking at, you know, kind of just broad general biogeographic trends in genetic diversity, looking at different natural environmental factors like energy availability and heterogeneity. And, you know, we found trends that were at least hypothesized for mammals, but in amphibians, just like, I think it was only elevation out of all of those environmental variables that actually had any relationship with genetic diversity. So something kind of weird is going on with amphibians. I wonder if it could also be related to their like unstable temporal population sizes compared to other taxa, but I don't know. So they're kind of unpredictable to me anyway.
Okay, great. So if there are no more questions, oh wait, we have one more. Yeah, go for it.
Sorry, squeeze one more. It's a bit out there, but have you looked at how your patterns of genetic diversity correspond to species level diversity measures at all?
Yes, I had a paper come out earlier this year about just that in evolution. So if you want to take a look, you can. The brief story about that is there doesn't seem to be a strong latitudinal gradient in genetic diversity, and genetic diversity, at least in North America, seems to be negatively related to species richness, which was not what we expected to see. And that was true in mammals, and also I have a preprint looking at that in amphibians, and also looks, the spatial patterns are opposite. The environments that cause that, I'm less sure, because as I said, amphibians don't like to play by the rules, but yes.
Cool, all right, thanks. I'll go check it out.
Okay, fantastic. So I think then if that's everything from everyone, please join me in thanking Chloe again. Chloe, it was great to have you. That was super interesting. And like I said, yeah, claps, claps, claps. If anyone has any questions for Chloe, you can email her, or you can email me to get her email address if you need that. And yeah, thank you again.
Thanks for having me.
It's our pleasure.
Thank you for the very lively discussion, guys. It was a pleasure and very exciting. So I will see you all next week, or again online. I will send an email reminder, but that's gonna be like that. And if you want the recording from this session, or if you hear of anyone that would like to see Chloe's presentation, please contact me. I will be able to share it. So I'm gonna actually stop it now.